Okay, I'd like to thank the organisers for the invitation to speak here today, and as you can see, I don't have a French accent. I toyed with the idea of trying a French accent, but didn't think I could carry it off for a full 20 minutes. I'm going to be speaking about tissue reservoirs today, and my talk will involve discussing what a tissue reservoir is, the types of tissue reservoirs, and in particular the central nervous system as a tissue reservoir, and the impact of the central nervous system on eradication and eradication on the central nervous system. And also, I'll try to incorporate some highlights from the NeuroAIDS pre-conference symposium that was held on Friday at AMREP. So, there are a number of tissue reservoirs that have been identified. Of course, the most significant of which occur in the gut-associated lymphoid tissue and the lymph node. And a number of studies have looked at this tissue reservoir extensively, and basically what they've shown and what they agree on is that within several regions of the tissue from the gelt, they carry significantly more HIV DNA than in blood. In a study which has been shown in a poster by Jenny Anderson from Sharon Lewin's group, they looked again at the distribution of integrated DNA or latent integrated DNA, and what they found was that was preferential infection of rectal CD4 positive T cells compared to blood and lymph node CD4 positive T cells. And when they looked at the distribution or which subset of cells this DNA was found in, they found that there was preferential infection of central memory CD4 positive T cells in the blood that expressed both CCR6 and CXCR3. They then looked at the distribution of these subsets within rectal tissue, and they found that rectal tissue harbours a high percentage of memory CD4 positive T cells expressing both CCR6 and CXCR3. And when this is taken together with the finding that in blood, memory CD4 positive T cells expressing both CCR6 and CXCR3, that they also harbour more or increased levels of DNA, this may explain why rectal tissue has a larger integrated DNA reservoir than blood. There's also been reservoirs identified in respiratory tissue, the kidney, the reproductive tract, and as I said, the central nervous system. And this is where I'll spend the remainder of my talk. So the CNS as a viral reservoir, it's a potential but somewhat controversial reservoir of HIV. It's potential because during the course of disease, HIV enters the central nervous system, it replicates and it can cause disease. It's controversial because there is really no direct evidence that in a virally suppressed patient, the CNS contains HIV-infected cells which harbour a competent proviral genome. However, there is a significant amount of indirect evidence from numerous studies to suggest that at least in some HIV-infected patients or virally suppressed patients, the CNS could present a viral reservoir. So there are key questions relevant to the CNS as a compartment, as a tissue reservoir. Are cells of the CNS infected with HIV? Are they infected in sufficient numbers and contain proviral integrated genomes during suppressive heart? Is there evidence of ongoing persistence and replication, immune activation during suppressive heart? And do these cells have mechanisms which allow them to become latent? Are they long-lived cells? And are there specific mechanisms which facilitate a latent infection? So if we look firstly at whether or not they have integrated genomes, the CNS targets of infection are macrophages, and these are the major sites of productive infection, and astrocytes. And astrocytes have a non-productive and restricted infection. So to determine whether or not cells have an integrated proviral genome, the only data that we have is from symptomatic patients. In 2006, we showed using laser capture microdissection from autopsy brain tissues where we purified pure astrocytes, macrophages and microglia, and we identified integrated genomes using LU-PCR. We showed that proviral genomes could be detected in macrophages, microglia and astrocytes, but not in neurons. We also looked at the extent of infection and to determine whether there were substantial numbers of cells that could be infected. Again, we used laser capture microdissection, and we looked at the envelope sequences in astrocytes and macrophages. This time we used asymptomatic patients, And basically what we showed was that when you purify these cells from different regions of the brain, C being macrophages and astrocytes isolated from the perivascular region, and B away from the perivascular region, and A being the deep white matter, you can see that you could detect astrocytes and macrophages in all of these regions, and the level of detection increased as you move closer to the perivascular region. And we failed to detect, again, any infection of neurons. This has also been confirmed in pre-symptomatic patients by Thompson et al. In 2011, again using laser capture microdissection, but this time with pre-symptomatic patients, and they detected HIV DNA in P24 negative perivascular macrophages, microglial cells and astrocytes. They also showed ongoing astrogliosis and microglial cell activation in these patient tissues. There's also evidence from the fact that we have distinct evolution of HIV. So the HIV is compartmentalised, not only between the CNS and non-CNS, but also in different brain regions and different cell types within the brain. This compartment, that's at the level of envelope, NEF, the LTR and TAT. There's also a functional compartmentalisation. CNS-derived envelopes have an enhanced ability to utilise low levels of CD4, and this permits M-tropism. This basically results in a more open conformation which exposes the CD4 binding domain and an increase in the sensitivity to CD4 binding site neutralising antibodies than matched envelopes that are isolated from the blood of these patients. So is there ongoing replication in the CNS in the presence of CART? Well, there have been reports of asymptomatic CSF viral persistence. In 2010, Eden et al. Looked at patients on suppressive CART, and they showed that in 10% of these patients there was detectable viral load in the CSF using a standard assay for detection. And as part of the Charter cohort, Scott Latonda showed that in 300 patients, on suppressive heart, that 40% of these patients had a CSF viral escape with more than 2 copies per mil in the CSF. This is further supported by numerous reports at CROI this year of asymptomatic CSF viral persistence. Earlier this year, a study from Sarah Palmer's group where they followed 2 patients which had been initiated on heart, and what they saw was that when they looked at the virus from the CSF and the plasma, that there was a distinct compartmentalisation of viral sequences, suggesting that one of these sequences may have come from the CSF. There's also indirect evidence of ongoing replication in symptomatic CSF viral escape, and this is a very rare phenomenon, but it does occur. In this study, patients were followed, or in particular this patient 2000, followed for 8 years on suppressive heart with counts of less than 50 copies per mil. At year 8, they presented with the onset of HAN. They were still virally suppressed in the plasma. And what they found was that there were elevated levels of virus in the CSF and a significant level of pleiotrosis. The genotyping of the CSF virus revealed that there was resistance to almost all of the drug regime that was suppressing the virus in the plasma, and this suggested that there was ongoing replication evolution in the CNS away from the plasma virus. We can also look at immune activation and biomarkers for indications of ongoing CNS replication. In 2014, Crott et al. Looking at the presence of light chain subunit of neurofilament protein in the CSF, and this is a major structural component of myelinated axons, and it's actually an indicator of neuronal damage. The blue dots indicate no heart, the yellow dots are heart-treated patients, and the black dots are negative controls. And although significantly lower in the CART patients than in the untreated controls, NFL was still significantly elevated over uninfected controls, and this was suggestive of ongoing axonal injury. We can look at neopterin. Neopterin is a marker of macrophage microglia activation. It's an indicator of immune activation in both the blood and in the CNS. Levels of CSF neopterin normally correlate with CSF copies or RNA copies. Patients that were followed for 4 years of treatment with full viral suppression in the plasma showed that the plasma levels of neopterin returned to levels similar to uninfected controls. However, the CSF levels failed to resolve. This is also supported by imaging studies looking at increased brain PK1195 uptake. This is a marker specific for activated microglia in 7 subjects on suppressive heart, and also the excess of activated microglia CD68 positive cells in virally suppressed patients as well. So is there latency in the CNS? Do we know if there's latency, even if there is ongoing replication? The CNS infection of astrocytes, just by its nature, is a latent infection. And this latency, or this block in transcription, this major block has been measured at the level of transcription. However, in vitro studies have shown that astrocytes can be infected. They can be carried in tissue culture for several months, and these are primary fetal astrocytes. And then you can have activation using TNF-alpha and interleukin beta. And in line with some of Corinne's earlier comments, a recent finding showed that in HIV-positive autopsy brain tissues, where there's HIV-positive latent microglia, there's elevated levels of the CTIP2 protein, which has been shown to be involved in suppression of transcription. We can also look at the animal models in this study by Christine Zink and Janice Clements. Monkeys were inoculated with a neurostrain of SIV. They were treated or not treated at day 12, and the CNS and other tissues examined. And what they found was that despite a decrease in HIV RNA and GP41 in the CNS, there was no decrease in the level of HIV DNA following treatment. So, if we do have an ongoing persistent infection in the CNS, what is the effect of the transcriptional activators that are currently being used in the periphery or in T cells to activate virus? To look at this, we did this in collaboration with Sharon Lewin at the Burnett, and we looked at a range of transcriptional activators, and as you can see, they have a varying range of CNS or predicted CNS penetrance. We initially looked at the toxicity of these on different cells. We used macrophage-derived MDMs as a type of macrophage or brain macrophage, and primary fetal astrocytes. And as you can see, they were relatively well tolerated in all of the cell types, with the exception of chiatocin and romidepsin. Both were very toxic to the PFAs. In order to analyse the effect of these activators, we needed to introduce the LTR into our primary cells. And to do this, we used a vector that was generated by Damien Purcell and Jonathan Jacobson at the Peter Doherty. And basically, the vector is a full-length genome with deletions in GAG, POL, and envelope. It has a luciferase inserted into the NEF open reading frame, and you can introduce the LTR. These are delivered, the LTR is delivered to these primary cells following generation of a VSV pseudotype virus. We went on to show that any activity or any luciferase activity was dependent on integration using raltegravir. And basically what we found using the various transcription activators is that romidepsin, JQ1 and pubinestat were the most potent, as you can see, in the PFAs. There was some activation in the MDMs, but it was very minor. So, in summary, there's strong data to suggest persistence in the CNS during persistent heart. The CNS by nature presents unique and extensive challenges in terms of eradication. How do we treat activation in the CNS? What are our targets? Do antiretrovirals penetrate the CNS, and do they function in the CNS? Can we rely on immune control? Do we need more biomarkers? There are very limited biomarkers for looking at activation of virus in the CNS. And assuming the CNS reservoir exists, current cure strategies are likely to result in replication in the CNS. Tissue reservoirs are currently the major barrier to eradication and cure of HIV-1. Identification and characterisation of all these reservoirs, including the CNS, is critical. And importantly, we do need to show the cells within the CNS during suppressive CART contain a competent integrated genome. I'd just like to thank the people who worked on this project, the people from my lab, especially Lachlan Gray and Wan Jung. We work closely with Paul Gorry. We have a very strong collaboration with Sharon Lewin and Damien Purcell. I'd like to thank the funding bodies NHMRC, the NIH, and the Delaney AIDS Research Enterprise. Thank you.